So my name is Hideki Ueno from Kyoto University, and I'm really happy to be here and to give a talk. But my talk only has 10 minutes, and plus maybe alpha for discussions. So I really simplify my talk today. So I want to talk about only one thing, which is high-avidity T cells, of CD4 T cells in humans. So I want to really go back to basics, so I got it from a textbook. So basically, I want to talk about the T cell antigen specificity. So the naive T cells, if you look at it, they're so diverse, and only the antigen-specific T cell become selected during the priming phase, and they become clonally expanded. And so in this case, so this is a kind of B cell, but the T cell is exactly the same. So the clonal expansion, selection of the clonal expansion is really key of the adaptive immune response. Another key is that memory T cell, B cell can make T cell and B cell can make a memory response. So once they are primed, so they take a little bit time to grow up, but they become effector cells. And some T cells remain as a memory cells, and after second stimulations, they really grow rapidly, proliferate, and they do something good to protect the body, right? That's a really immune response. So in that sense, memory response can be defined as kind of faster, the more magnitude response. So in meaning that really T cells, by talking about the antigen-specific T cells, that includes naive T cells, memory T cells, different subsets, and we want to understand what's going on when we talk about the antigen-specific T cell response. So that's why I want to propose, basically there are three key parameters to assess the antigen-specific T cell response in humans. So one is, of course, the frequency, so magnitude. So, of course, if you don't have so many T cells which can react to some antigens, you cannot expect much kind of strong response, right? So the frequency really matters. Second parameter, quality. I'm really like a CD4 T cells. So there are many CD4 T subsets, right? Like a TH1 cell, TH2 cell, TH70 cell, TFH cell, Treg, so on. And also in the case in the CD8 T cell, there's activated T cell, exhausted T cells. That's also the case for CD4 T cells. So we need to understand the status of the T cell subsets as well, right? And so here what I want to propose today is avidity is another really key compartment of the T avidity. It's a T cell avidity against a peptide MC complex. And so this is really key parameters to define the sensitiveness of the T cells against like a peptide MC class 2 or class 1 complex APCs or target cells. So the question, how do we define avidity in a kind of really good platform? And basically what we found, so published in 2019, is that the IRF4, which is a transcription factor, is a really good indicator to identify the avidity of the T cells. The reason why that's because when T cell receives T cell signals, via recognition of peptide class 2 complex or class 1 complex, so if this T cell signal is really strong, that spontaneously products a very large amount of IRF4 proteins. So if you have a very low avidity T cells, the T cell generate only low amount of the IRF4. So by measuring the protein IRF4, basically you can measure the avidity of the T cells against the given the proteins. So by using that, basically we try to understand the T cell antigen-specific T cell response in a kind of many kind of cases, including like in uninfected, unvaccinated, kind of cross-reactive T cells, and also vaccinated after vaccine response. And also we deal with many kind of COVID-19 convalescent patient samples, and also we are dealing with the long COVID patient samples to really see what kind of differences of the antigen-specific T cell responses in a kind of different individuals. So we are kind of really going very diverse and comprehensive manners by using both FACS and the single-cell RNA sequencing. And also our favorite is use these trimeric spike proteins in the case of spike-specific T cells. Because by putting this spike protein to the PBMCs, so PBMCs, some APC capture the antigens and naturally process the antigens to the peptides. So what T cells see is a naturally processed peptide and the complex with the MSC molecules. So we basically like to use these trimeric spike proteins. And also to look at the cross-reactivity, we deal with trimeric H-CoV-derived spike proteins as well. And we culture for 24 hours, and we get activated T cells, and we analyze 32 color flow cytometry to really define the T cell subsets. And also we sort the disactivated T cells so that we can do single-cell sequencing, plus T cell sequencing. So the basic, I think that you are very familiar with this kind of technology. It's called AIM assay, it's activation induced mark assay, that we use also to in this assay. So we culture the PBMC with thalamic proteins, and that's processed by the APCs inside the PBMCs. And when there's a T cells recognize this peptide class 2, class 1 complexes, T cells become activated, they express activation markers. And we use these 3 markers for the case of CD4 T cells, CD25, OX40, and CD69. And also we analyze CD8 T cell activations by using CD25, CD137, CD69. But in addition to it, we want to see the avidity, right? So that's why we look at the intranucleus IL-4 protein level to detect the avidity of the T cells. Actually, that works very, very well. So here I'm showing one example of the comparison between the uninfected individuals versus COVID-19 basically infected patients. So we use COVID-19 spike proteins. So like you see, so this is really what you see. Like if you use a blood sample from COVID-19 convalescent patients, you can clearly see these isolated populations expressing high level IL-4 and ICOS. This is high-avidity T cells. And in the case of uninfected, it's kind of cross-reactive T cells, basically majority lacks these big populations. And because we have many, many markers, we can really characterize kind of what kind of marker they express, but it's really going into detail, so I'm going to cut today. So the reason why we're saying that they can be high avidity is that because they can be really sensitive to the protein expressions, protein amounts. So here we titrated the amount of spike protein in the PBMC culture. We are usually with 5 microgram per ml. But as you see, if you decrease spike protein inside the PBMC culture, you tend to kind of increase the populations of the high avidity T cells, meaning that these high avidity T cells are more sensitive to the antigens. They require much less antigens to be activated. And also we wanted to confirm that this is a really high avidity by looking at T cell single-cell RNA sequencing. So as you see, this is completely isolated populations, which is an IL-4 high population, expressing IL-4 high level. So by looking at the gene profile, basically we found, so this is a case with the COVID-19 convalescent patient samples. We found these IL-4 high populations are largely composed of TFH cells, T follicular helper cells, and the TH1 cells. And of course, we can see kind of other subsets inside these low-affinity T cells, including Tregs, inflammatory cells, and CD4 CTLs, and weakly activated, like very similar to central memory T cells. And interesting that so we did this kind of thing by using FACS and to see what kind of recompositions that each patient made after the COVID-19 infections. So here I'm showing the kind of difference between the female and male patients and among the mild disease, moderate disease, or severe disease patients. So here we are looking at the composition of the different subsets within this antigen-specific CD4 T cells. So as you see, particularly in the male much more dominant inflammatory T cell subsets, and much less Tregs, much less CD4 CTLs, and IL-4 high avidity T cells. Much more like a T cell response, much more skewed in the male very severe patients. And the funny thing is that it's not only COVID-2 spike protein specific T cells. So if you do the same thing with AHCOVs, with human coronaviruses, like OC43 or NF63 spike proteins, basically you see exactly the same thing. So male patients, basically when they get severe, there's a very clear-cut skewing of the T cell subsets, much more inflammatory and much less regular T cells, and much less high-avidity T cells. So it's really indicating the kind of power to analyze avidity of the T cells to understand disease pathogenesis. So I want to move on to the vaccine studies and to see what kind of response can be seen by using this technology. So here we basically take 32 donors. Sorry, I need to go back. Yeah, 32 donors, and they include many female, males, young and old, many diseases. And we get the samples before and after first and second vaccination of the RNA of the COVID-19 vaccines. we get the blood samples, and basically we look at the COVID-2 spike protein specific CD4 T cell response. In the case, basically by fax. And also we analyze antibody titers using Luminex and neutralizing antibody titers. So here I'm showing the one example. This is after two shots vaccinations. So by using IL-4, so here we get it to antigen-specific activated T cells. You see IL-4 high population is clearly visible among these high-affinity, antigen-specific CD4 T cells. So by looking at the kinetics, so this can be shown here. It's the first boost after first shot, but beautifully boosted after the second shot. And then went back to the 6 months after the second shot. So actually there's a really pretty much heterogeneity among the individuals who responds well to the vaccines and who poorly respond. And then basically we split into 3 groups. One group is a very good responders, where we could see high-avidity T cell expansions after second dose. So this is a moderate. Somehow we could see some expansion, but not much. So and the weak responders. And as you expect, there's a kind of clear-cut kind of difference in the antibody titers. So strong antibody responders basically make a kind of very strong antibody response, including other variants of the COVID-19 strains. But the important thing is that, again, by looking at the T cell subset within this high-avidity T cell at the post-1, so the majority is composed TFH1 and TFH1 cells, and in the fax. But the important thing is that by looking at this frequency, COVID-19 specific TFH cells, high-avidity cells, basically these populations, this beautifully correlates with neutral antibodies against the post-2 vaccines, and also durable antibody response, meaning 6 months after the second shot. Nice positive response. So I stop here. Basically, what we concluded is that COVID-19 induced COVID-2 specific CD4 T cells, and a fraction of which are high avidity. And high avidity CD4 T cells are composed of largely TH1 TFH cells, and all males do not strongly induce high avidity COVID-2 specific CD4 T cells, which may be associated with the poor clinical outcomes. And in the case of vaccines, early induction of high avidity TFH cells predicts durable antibody responses. So I stop here. Now I want to thank many lab members who contributed to this high-avidity T cell studies. And so actually we have 3 posters here, including those, the Rick-san and Serin-san and Maso-san. So please go to there and to discuss about this high-avidity T cells. And also thank these medical doctors, and also Hashiguchi sensei for making lots of trimeric spike proteins. So I stop here. Happy to take any question. Thank you.
Great talk. So in the male patient with severe COVID-19, they had abundant inflammatory CD4 T cell. So they expressed the CCR6. So I'm curious, this inflammatory CD4 T cell, they are more TH17-like cell or not?
Yeah, actually they do express much more like a lymphotoxin alpha, beta, plus IL-22, but not much IL-17.
For your low avidity T cells, so those that are IIF4 negative, low, how sure are you that they're actually even antigen specific? Because you gauge them with the AIM marker, but there's a lot of bystander activation. So maybe those are actually not antigen specific T cells?
That's a good question. So we try to really understand really the specificity. So because we do have a bunch of the T cell sequencing data, so we are making clones of the TCR carrying the same clones for the high avidity, low avidity to make sure they react. But in the case of like COVID-19 vaccines, so we know that the low avidity T cells get expanded, but not as much as high avidity T cells. So I think and also we can see the kind of expansion of the T cell sequencing as well. So somehow they look like they are responding, but as compared to high avidity, much less weak. So I think you're right. So basically, people are using the AIM assay to define the antigen-specific T cells, right? But whether that could be really bystander or not, because we are really basically using 4 markers, right? CD69, OX40, CD25, still kind of much less chance compared to only 1 or 2 markers to pick up the only bystander activation markers. But still, I think we need kind of evidence.
Hey, nice talk. I have two questions. The first one is, do you have a chance to have access to like human lymphoid organ like tonsil? And I just wonder what those high-avidity CD4 T cell would be like, germinal center, GC-TFH or non-GC-TFH? What do you have any thought?
So they are far from germinal center TFH.
Right.
But what are the questions? What
that's the first question.
Okay.
So they are non-GC-TFH.
They're totally different from germinal center TFH.
And the second question is, I just wonder if you stimulate your cell with current variants antigens, who will respond better, the high-avidity or low-avidity one? Thank you.
Yeah, so I think still we are working on that aspect to see, like the response to human coronavirus or variants, and there's kind of tendency when they make a good high-avidity T cell response, they also tend to induce a response against the kind of wider kind of repertoire of the coronaviruses, but still we don't know why.
Okay, thank you, Prof. Ueno, for the interesting talk. We know that the IRF4 is important also in B cells and multiple myeloma. So I'd like to ask, what is the actual function of IRF4 in the case of T cells?
Right. That's a good question. So actually, IRF4 does a bunch of bad thing as well to the T cells. Like if you have too much IRF4, basically that drive to T cells much more towards effector cells rather than memory cells. So really interesting for us is that this tend to be effector cells, but we know that these high-avidity T cells can remain as a memory cells. So still mouse immunology knowledge is not really enough to understand human immunology, and IL-4 in the CD4 T cell, that's really many things. Like if you look at IL-4, basically in the mouse, like you don't generate like a TH1, TH2, TFH cells. So that's so fundamental. So basically here, but we are not really asking what IL-4 does to the T cells. So we are using IL-4 as kind of tool to determine the avidity of the T cells.